Try to understand this carefully. This one. This is all for variety 1. So first variety under treatment 1 has the mean flag leaf area 14.4, 18.7 for under treatment 2, under treatment 3, under treatment 4. We have applied a different type of treatment and there is a difference. You can say 14, 18, 22, 16 and 17. Now either this mean is significant, these means are significantly different or not. To know this, we have to apply analysis of variance. This is the first expression. This is called description. Description of your data for variety 1, 10, 2, 3, 4, all of your varieties under different expressions have been shown here. That is another for temperature. That is the other variable such as tillers. You can check all the expression of all the variables. So after checking this expression, how will you go for analysis? Simply statistics, linear models. I will allow you to take notes for this. Write down statistics, linear models, analysis of variance, and then general ANOVA. This is the major path for the publication purpose. That is the first step to analyze the variation and variation. So click this. Now here, again, you will select all the dependent variables. Okay. Now you have to build a model. Model mean the independent variable variation. So for example, in flag leaf area or any other dependent variable from flag leaf area to this one, we want to check the variation in flag leaf area due to field. Click it. We want to check the variation flag leaf area due to genotype. Okay. Flag leaf area variation due to treatment. Okay. Flag leaf area variation due to reps. This is very, very simple model without interaction. Just we are dealing with our single factor analysis at a time. So all these factors are taken and we are not studying at the first place their interaction. We are not studying their interaction. Let's see what our model says about all these dependent variables at a time. The factor treatment is okay. The treatment is not, treatment has different levels. So we will go for like this. We can, for the time being, we can remove treatment. Let's see. Now, you can understand all the 8 analysis of variance tables are there. How you will interpret, how you will convert this into Excel, you will take this into Excel format and generate a publishable ANOVA table like I can show you on the screen. For example, we can have different ANOVA tables here for different work. You will convert this like so. From this output of 8 ANOVA tables, you can see that is statistics 8.1, that is date, that is time, and that is the variable name. So this is first factor. Due to field, 3 degree of freedom. 2 degree of freedom means we have 3 fields. 3 minus 1, that is 2. Sum of square is this. Mean square is this. Basically, when you divide this by this, you will get this. And the F value is 16 and P value is very much smaller than 0.05. It means the change in flag leaf area. This is interpretation. You should understand it carefully. I repeat, flag leaf area is significantly affected by the field. So there are three fields, 1, 2, and 3. The mean value of flag leaf area in field 1 as well as field 2 as well as field 3 is significantly different. So flag leaf area is affected significantly by field. Why we are saying that? Because a p-value is smaller than 0.05. So we have 11 genotypes in our experiment. 11 minus 1 is 10. That is degree of freedom, always total number minus 1. Total there are 11 genotypes. 11 minus 1 is 10. So this sum of square, when you divide this to this, you will get 662. Then f value, we can get f value by dividing. You can do it manually by dividing this with this. That is error. When you divide this with this, you will get 9.11. So this is also significant. What does that mean? Flag leaf area in our 11 varieties is significantly different. So we have genotypically different flag leaf area. Okay, this is the error. This is CV. CV is very much greater of this experiment. It has to be eradicated or some terms to be changed. This CV is not acceptable. You have to get under your agricultural experiments. If the CV is like this, 3, that is also very much different. So the tiller, this variable, and this variable, this data is very much fluctuated. So this CV shows that this data is not very much reliable. This data is not very much reliable. So there is a chance of variation due to any other factor that is uncontrollable, that remained unnoticed. So these two variables, we will not surely go with these until this CV is lowered down by eradicating that malicious factor. Okay. Now this one is fine. Temperature 3.49 CV, and only in the field the temperature is different, but in the genotypes TEMP variable is not significantly different. So for plant height, that is little bit acceptable. CV is 23. So the plant height has having the same results like flag leaf area. Plant height is significantly different in field as well as in genotypes. So this was the first expression to check. Now you will copy them all, for example, like this, and paste them at your Excel file like this. Now, see very carefully. You will go to the data tab, text to column. I repeat, after pasting your results from statistics to Excel, you will go to data tab, text to column, and then del F fixed width. Okay. You will select delimited and you will select space. Yes, now you can see every required column is divided. Go to next and then finish. Now you have separate in column your all the data. We need this one, mean squares, only for flag leaf area. From this, you will copy this, and here is 0.001. Like this, we had our results. You can convert it like this. You can convert it into text such as 0.00, as we have seen in our experiment, in our analysis. We can convert it also into text, not 0.01, 0.0001. We need this. Sometime you apply asterisks. Sometime if your all the data is significant, you go with it. You only select this, and you will convert this whole 8 different data into this publishable format. This is final publishable format of your data. You are going with percentage of variation in seed traits explained by 2 factor, 3 factor, or 4 factor. And these are your tables. That was your first table, for example, second table, third ANOVA table, 4 ANOVA table. So from collecting those ANOVA tables in Excel file in an expanded form, this is an expanded form, you have to compile them like this. How will you do that? See, if we reduce that screen, you can see, for example, this is weight or any other variable. You will check this field. Field will be like variety or treatment. These are your dependent variables. From here, you can write field, one variable in one row. And that row is this one from field, this one from genotype, this one from reps, and this one from error. So this column will be shifted into first row, and the name will be flag leaf area. So how will you do that? Simple. You will write here flag leaf area. You will write trait. You will write field. You will write gen. For example, you will write reps. You will write error, mean, grand mean, that is GM, and CV percent. Now what will you do? For this flag leaf area, you will copy this and click it and right click and paste it by opting this one. Now, this column is pasted in this row. Only this one you need. By saying this, you can place asterisks on these. For example, genotype is significant. You can apply 3 asterisks. 1, 2, 3. Okay. Field is significant. Genotype is significant. You can apply asterisks like this. Error, on the basis of error, you check these significant or not. Write the GM, that is 19.6. Write the CV, that is 43.5. So your all the table is shifted in a single row of the variable. So if you have 8 variables, you will just go with 8 rows. So this, in this form, you will have a single data, publishable data table from number one, you will take your raw data into the statistics. You will check your data feasibility for analysis, and then you will analyze your data for analysis of variance. You will copy your results to the Excel, and you will convert that data into different columns, set that data, and you will pick the most appropriate column like this. And this table, as you will generate on the basis of a number of variables, 1, 2, 3, 4, 5, 6, field, genotype, these are your factors. Any one factor, you can have treatment here, you can have field here, genotypes here, you can have salinity purpose here, you can any other factor variables, independent variables here. But the things to note are, you are going to present one variable in one row at a time when you are going to publish your data in thesis or in your research paper.